Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Good evening, I'm Yvonne Stapp for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. Tonight we learn about one of the big challenges in evolutionary science, the effort to reconstruct ancient genes to learn how they adapted over millions of years. Our guest is Betul Kacar, a research fellow and astrobiologist at Harvard University in the Department of Organismic and Evolutionary Biology. Dr. Kacar is also Global Science Coordinator of the Earth Life Science Institute at the Tokyo Institute of Technology and co-founder of SaganNet.org, an online astrobiology grassroots network. Dr. Kacar received her Ph.D. in biomolecular chemistry in 2010 from Emory University. Before taking her position at Harvard, she was a postdoc fellow at NASA's Astrobiology Institute, where she combined experimental paleogenetics with laboratory evolution. More about that tonight. Dr. Kacar has received considerable recognition for her innovative approach to this work, and tonight she explains how she revived a particular ancient protein, then got it to evolve inside a living microbe. This was thought to be nearly impossible, so we'll likely hear a great deal about her work in the future as this methodology develops. Tonight we'll learn from Dr. Kacar why scientists are trying to reconstruct ancient genes and what that work will reveal about evolutionary processes and possibly what it adds to astrobiology. We're very pleased to welcome Dr. Betul Kacar. Welcome, Dr. Kacar.
Thanks for having me.
You're very welcome, and I'd like to start by finding out what this field is first off, because apparently it's very new. Could you give us a little background on just the field itself?
Thanks for having me. I will be talking about two seemingly distinct fields as my research combines these two aspects. One is experimental evolution, which is really the ability to evolve organisms, and these could be bacteria, this could be yeast. Particularly, I'm talking about microorganisms in the lab with the conditions that we set up and define in advance.
Right.
And the other field that my research benefits from is the paleogenetics field, namely ancestral sequence reconstruction, where we access the ancient sequences of contemporary genes and proteins through inferences of phylogenetic trees.
Right. Now, I understand this is very new. How new, roughly, this paleogenetics?
Well, as many aspects in science, as many fields, the roots go back to around 1960s.
Oh, okay.
Where this approach was proposed as an idea by a chemist and a physicist named Emil Zuckerkandl and Linus Pauling. They published this paper that suggested that perhaps we can take what is currently existing and then use that as a tool to reconstruct our way back in the past and access the ancient sequences. And keep in mind that this time they proposed this as an idea. And the paper beautifully outlines the figures and here's how one could do it. And they perhaps foresaw that maybe 20 years later we will have the approach of synthetic biology where we can actually turn their idea into reality.
Right.
And in the late 80s, early 90s, researchers including Steve Benner took this idea.
He wouldn't he? Yes, that's right.
and actually reconstructed an ancestral protein for the first time. And since then, the field has blossomed, I would say, if one takes the time and looks at the papers published on the subject matter, you will see a rapid development, especially the last 5-6 years, I would say. Has been like, there's been a great deal of interest into field and more papers coming out.
So you're one of the pioneers in developing, especially what you're getting into, what you're going to be describing for us tonight, which has gotten quite a bit of attention, and I'm sure we're going to hear much more even later. So the field is fairly new, and what's interesting just listening to you is that it gets started with physics and chemistry, not biology, but all of these things are merging, but it's interesting how much comes from physics and chemistry. Is there any reason for that?
I would say maybe it's also one way to think about it would be to think that it's maybe not narrowed down to the discipline, but perhaps the vision of the scientists. Maybe once you start seeing no barriers, but maybe once the barriers disappear between different disciplines, you can see things more clearly and coherently. So maybe the fact that Emil Zuckerkandl or Linus Pauling were scientists that really didn't see science perhaps in a box as physics or
they were
chemistry.
already outside the box.
Exactly. So I think it takes outliers really that will come up with these revolutionary ideas, and another outlier to see what they're proposing, like in the case of Steve Banner, and really go for it. I will talk about experimental evolution field more. But the one of the pioneers and really the scientists who really I think made it all possible for us right now. One of them is Richard Lenski. I'm lucky to have conversations with him every now and then, and he always says that the literature is full of gems, and one should really go back to early 1900s even, and really find the old papers where people suggest these experiments, the hypothetical themes, and a young scientist can actually turn that into maybe something really happening. And maybe it could be because maybe back then there were less boundaries, or people maybe didn't think.
I guess maybe it got very narrowed, the academe, just research and stuff.
Maybe take now your career depends on the results of the experiments, and you don't want to take big risk. You want to do something new and pioneering and exciting, but not too new that will fail and you will disappear from the
That's a very good
pages
point,
of science.
because it comes up again and again and again that, especially with funding cutbacks and stuff, that scientists today fear taking chances, where in fact the nature of science is to take chances, and that you need to be able to fail. You need to say, well, okay, that didn't work, got to start over again. So it's curious that we're having this moment. I hope we get over it, and certainly, in your time, I would like to go to your work now, which has been so exciting. Can you give us an idea? First of all, you're going to reconstruct something. Tell us how you do this, if you would, please. So,
even though the name implies ancient genes, I do not study fossils. But I use DNA as a proxy, and I treat the currently existing DNA as a fossil. At the end of the day, it is what's remnant of the past, and it is right in front of our eyes. So the whole approach, the paleogenetics approach, follows a backwards from present methodology, where we first collect the DNA sequences from currently existing organisms. We take the gene variant or protein sequence from you, from me, from a crocodile, from a flower, and then we look at the differences and similarities between these sequences.
Could I ask, do you take just any old gene or protein, any old gene? Are there some that are conserved over eons and others that are newer or lost?
That's a very interesting point, because usually this is an overgeneralization, and I'm sure there are exceptions to this, but the genes that are conserved tend to be older. Essential genes are several times shown to evolve slowly, for the obvious reason that if there is a mutation there, the consequences could be tremendous for the cell, and we need to keep the essential components.
Then are those genes represented across organisms in
Another good point. Yes, usually the genes that are important for a cell's function are conserved in different homologs.
It doesn't even matter what the organism is, plant, animal, insect, or whatever.
Exactly.
Is that the idea? It just has to be in the cell?
Exactly. For example, when we look at we have two different sets of genes. We call these informational genes. So for example, the cell making, the DNA protein making machinery of the cell, ribosome, is one of the information processing centers.
Yeah.
So you can treat DNA as information, and then ribosome constantly reads through this information and tries to make sense of it, and then produces proteins. If the protein doesn't make sense, usually the cell has a mechanism of eliminating this wrongly translated, mistakenly translated protein. So this is information.
Right.
And there are housekeeping genes. These are the genes that make sure that the house is kept safe. I would
because
say.
It's so complex inside that cell.
Yeah.
Okay.
So it is in a system of, everyone is equal, but some are more equal.
Okay.
In the cell.
That's familiar. Okay. All right. The elite is the elite ones.
Yeah.
Okay. All right. So then you select something that, in this case, it was a conserved one of these.
So I went to
conserved
the information center of the cell. So I went to the ribosome, which is the machinery that makes proteins, and I picked a protein that is very essential.
Yeah.
That takes care of a very important function for the cell. We need amino acids to produce proteins.
Right.
Well, by we I mean our ribosome, and this protein that I selected, which is called elongation factor, EFTU, or EF maybe for simpler purposes, is responsible from carrying the amino acids to the ribosome. So it really brings the food, I would say, to the processing center.
Okay.
And so we can think of it as like a shuttle protein.
Okay.
And so I went right into the core. It is known to be a very conserved protein in terms of itself. It is not very different if you look at different organisms, but also it is thought to be one of the oldest proteins in the cell that since the beginning of life or the last universal common ancestor that we call as LUCA is presumably had this protein in its machinery. Okay. So that's the one I picked.
Okay. Then the next step, so you select this particular one that is conserved across organisms, all organisms. You pluck that little guy out, and then you did what?
Well, you can imagine that I almost had invisible scissors. And well, in this case, actually, they were molecular scissors. I went into the genome. I cut out the currently existing, the modern gene, EF gene in my case. And then I replaced this with, it is 700 million year old, inferred to be 700 million year old ancestor.
Okay, you replaced it with the ancient one. Where did the ancient one come from?
Exactly, very good question. I took advantage of the ancestral sequence reconstruction methodology, where the EF protein was a product of a phylogenetic reconstruction that was already available to me.
All right. And so you take that, and then it starts to get very interesting, I think, if I understand
it.
Yes,
next week.
Things happen. Right?
I'll leave it here.
It's because this, so you take this guy out and you put it into another, was it into E. coli? Or
so this is all happening
in E. coli.
E. coli.
Is always everywhere in the labs, right?
So it's a very simple system in that sense. I went to E. coli. I removed E. coli's currently existing essential protein, and I replaced it with its 700 million year old ancestor. So upon this replacement, things did happen.
Yes, and this is the fun part now.
Fun for us, because we get to observe it, but not too much fun for bacteria, because bacteria suffered.
Right. We're used to taking a gene out of one thing and putting it into another.
Yes.
But getting it to evolve is a totally different thing, evidently, and this is what
so you say it really well. We are very used to taking a currently existing gene from organism A and putting that in organism B and see what happens. We're used to these exchanges.
Right.
It's funny, actually, I say it so casually because even that is very difficult and takes a lot of processing and really a lot of design in advance. And definitely very valuable way of understanding why a gene is there at first place. Just get rid of it, or get rid of it by replacing it with another, a neighbor, a cousin, basically, that exists in another organism.
Right.
So to complement those studies, I did say, okay, well, we can go and kind of travel in the current existing time.
Yes.
But how about we go back in time and go to the ancestor and replace this ancestor, place this ancestor inside modern microbe and see what it does.
And what did it?
Well, so before I did this, there were studies on this ancient component, biochemical studies. So these did not involve whole cell, but it did involve some selected components of the whole cell. So we had some idea of what this ancient component does in terms of interacting with some of the partners in the ribosome.
Right.
And these preliminary experiments showed us that this protein doesn't do that well compared to the wild type, which is coming from, which is what we say, the wild type.
Yes, right.
It's a funny way of saying
not
it.
Your lab.
That's our lab language. I don't know why it would make a nice rock band name. But it is it's the currently existing, did a lot better than the ancestor. Maybe this is not surprising.
Okay.
Because you may think that evolution may optimize some of the components in the molecular level.
Yes, we do assume that, right? But
so what I saw when I did this genomic level replacement was complementary to the biochemical results. That this presumably, maybe because in the ribosomal machinery, this ancient protein didn't do well anymore, and the whole cell grew slower than it did before.
So it had difficulty replicating, or it didn't replicate, or it did replicate, but it was weak, or?
It did grow, which was good news for me, because if it didn't grow, then I cannot work with a dead organism.
Right. Okay, that's right.
So I'm here now because it worked.
Right, you are here because it worked, right, okay.
But the idea was that it will be not perfect, but it will be viable enough. It will be efficient enough to produce a viable bacteria, which is what happened. So bacteria, unlike us, generates a new offspring every 20-25 minutes in the environments that I select, in the conditions that I provide, and for the bacteria that I work.
Right, right.
In this case, you can maybe roughly I can say that a bacteria that grows, that doubles every 20 minutes, started to double every 50 minutes.
I see. So it slowed down.
It slowed down.
Yeah.
And and then I subjected this suboptimal, I would say, organism into passages of laboratory evolution to see what will happen. How will this sick man stand up again?
Yes, right. But it did in the end. So we have multiple generations. Every 50 minutes we get a new generation of these bacteria, right? With this, what appeared to be a fragile gene at this ancient version of it.
That's one way of putting it, exactly.
Right. But what happened in the end then?
So this ancient protein triggered some sort of perturbation in the system, so that the system no longer grew as well as it did before. So something has changed.
Yeah.
And you can think that there are so many different ways to cause a problem in the cell, actually. You can create so many problems.
Pretty complicated, yeah.
But in this case, we really think we narrowed it down to one single component, which was the gene itself.
Right.
Right? So we went right into the information center of the system.
Right.
And then we replaced a very essential component with its divergent counterpart. Not so shockingly, the system didn't produce healthy offsprings as it did before.
So the cell itself had to make a major adjustment on the basis of having one new gene, one substitute gene.
That's one way, or you could think that, or maybe it will be subtle steps towards optimality.
I
so
see.
you can think of think of this in two ways. My instinct was also that the response to this such a dramatic change will be very drastic, and the system will want to stand up very quickly.
Yeah.
Because you cannot live like this.
Right, right.
And so experimental evolution at that time provided a very nice tool, which is, subject this organism to laboratory evolution, and then monitor the changes in the gene and the rest of the genome. See what happens in the cell, because you can think of 3,999 different genes that could respond to this ancient component.
Right. The organism didn't fare too well over some generations, but in the end, did it sort of, what would you say, revive? Was it, did it?
I'll take a few steps back again and maybe tell you what I was asking before I started this, at this moment when I put the ancient gene.
Yeah, right.
The idea that I was really trying to understand was the repeatability of evolution. So this is a very interesting question to me. Why are things the way they are?
Yeah.
Was there a molecular reason behind this? Could evolution repeat itself? And if so, what would we see? Are the systems, even we, are here for a particular reason? Maybe for the forces of determinism? Or is evolution just a fluke accident? So I was really interested in testing the dynamics between chance and necessity in evolution. Now that we...
Back to Mano.
Exactly. So the idea that was also proposed and was touched by Mano, but also biologists like Steve J. Gould proposed that we can treat everything that existed in life could be visualized as a recording. And let's assume that everything is recorded on a tape. So Steve J. Gould named this as tape of life. Of course, our young viewers who are hopefully watching,
much more digital.
They will not know what tape is.
What a tape, right.
But maybe you can show a picture of tape. This is what tape is. And so the idea was that what if we erased everything that was recorded on this tape and then replayed this tape again? Would we listen to the same tune, or would it be completely different? So this was what Steve J. Gould and many other biologists were interested in understanding. And philosophers like Daniel Dennett, who's here in Tufts, also tackled this in his book. And he wonderfully used this analogy where he said, we can think of this as a clash of dialects. So he argues that what if we think that everything that we see around us and everything that exists now has evolved their own particular dialect? So rewinding the tape would be almost erasing all the dialect and then expecting the same language to evolve. Or we can think of it as, instead of changing the whole dialect, we can change the dialect for one component and bring an ancient dialect and expect that ancient dialect to communicate with the currently existing system, which is really, I think, what I do, or how I want to see what I do. That we have the whole cell that has adopted a dialect, that the cell is communicating. There are networks, there are members in this network that's optimized to the current condition. But I am taking something that existed 700 million years ago. So it's an ancient dialect. And this component is an essential component. And from the analysis that we do, this component interacts with a lot of different things. And usually essential components they are older, they evolve slowly, and they have more interaction partners compared to the younger additions in the network. So you're changing a very core part of the whole network with an ancient dialect, and then expect this ancient dialect to maintain some kind of conversation with the offspring. And in this case, the language is molecular evolution, binding or different mutations or changing different subtle interactions. And I was interested in understanding this, that if I put this ancient dialect inside, will I revive and replay a whole new language
Right.
Or will the ancient protein accumulate mutations
Yeah.
that will resemble the same exact modern component?
Right.
Will I listen the same story again and again, or will I hear a completely different outcome?
Right. And I think this is like the first time that's really been done. What was the outcome?
So the outcome wasn't either of them. The cells surprised me.
Okay.
No, actually,
none of the above, or all
none
of the above,
or
or
all of both in some ways. So what happened is that, so I always thought that either the ancient protein will accumulate compensatory mutations and resemble a modern component, or the ancient component will accumulate very different set of mutations and will be a completely different protein. Not, I guess, completely different, but will be different than what I currently see.
Right, right.
And in which outcomes I thought, okay, these would still be restricted.
Right.
Because the system is really focusing on the major problem, which is the ancient component. Alternatively, which I hypothesized that this would be more chance outcome, that the changes that will accumulate in the system will not be fixated on the ancient component, but it will be elsewhere in the genome.
I see.
And it will be my job to figure out what those changes are. So the answer was the mutations did not accumulate on the ancient protein, and the mutations didn't accumulate on very different pathways either. Mutations mostly accumulated in the region that will regulate the amount of the ancient protein.
Oh.
So the protein didn't change,
Right.
but
Right.
how much of the protein is being made in the system did.
Yes.
Which was a clever way of, fixing this problem. And looking at the literature, there are so many examples that show that mutations that regulate protein or gene level in the cell are dominant forces underlying evolutionary innovations.
Oh, wow.
And in this system too, that at least for the time being that I generated in the lab, what I saw was don't alter the protein, but alter the amount. So don't try to make something better, but just make more of the suboptimal component. Interesting.
It's very interesting. What's the next step for you where does this take you now?
Well, we have more lineages to analyze in terms of we did evolve this populations in parallel in various amounts. So we want to understand what happened in our older generations. That's our first and foremost goal. Also, we are interested in extending the system to different genes and maybe networks that will be informative about how life evolved. Maybe 2 questions related to exobiology, astrobiology, the life's history and Earth's history.
Right. A couple of things. Would you expect differences with different genes? Is that's a possibility? So the outcome in this case might not necessarily be identical in another one. In other words, you might get different outcomes or something. Is that because you're dealing with very complex systems, which I thought was one of the things that you sort of underscored. This is really complex. Right. So this part isn't known yet. It's still new. May I ask about the astrobiology too? You're looking for life. Everybody's looking for life somewhere else. And so do you expect similar mechanisms? Life is life is life, or you have no clue? Or
one overarching question that my research had was to understand the degree of repeatability of life in the sense of understanding the presence of life elsewhere in the universe. So the question is, if we detect life, which I believe one day will, how would it look like? And before even detecting it, what are the likeliness of finding life elsewhere in the universe that resembles life we have on our planet? So those are the questions that really motivated me to get into the field of astrobiology, and also the reason I believe NASA generously funded my research for all these years.
Right.
We take, in my case, what we have here on our planet,
Right.
and really try to understand the underlying evolutionary dynamics of what we currently see.
Right.
We know so little.
It's so true, isn't it?
Yes,
All the time.
everything we do comes back to the notion of it's very complex.
Yeah, my sense is that convergence of biology, chemistry, physics, geology, all these things, you need a different frame of mind. Is that true? And do you see this going on now?
Yeah, so NASA has a very nice way of putting this, especially in the astrobiology field, that it is not interdisciplinary but transdisciplinary, that we want to really just create these bridges underneath and connect with each other.
Right.
That we don't want just to communicate, but really create these bridges. I think there's a big difference. So I think understanding questions like how life started, where is life going,
what it is.
You can't, exactly, right? So this is a question beyond biology, even though biology is at the core of understanding life.
Right.
So it is exciting that nowadays you can discuss the same question with a physicist, with a geologist.
It's so true. Yes, that's right.
And we are all getting together, understanding that, with the same question that unifies us.
Yeah.
So that's very exciting.
It must be, because I see this like with the Mars exploration thing. There's a huge community of scientists representing many different fields, and they put their heads together.
Exactly.
You know, their expertise gets combined. It just seems like a very new approach to many areas of science.
It's funny because, this is bringing us back to the beginning of our conversation where we were talking about Emil Zuckergandel's work, that we think maybe one reason that these pioneers came up with these brilliant ideas was because they didn't see any walls. There were no borders on their thinking.
Right.
And now maybe what will also be good in moving forward will be to revisit the roots of scientific knowledge, which is not to think that I am a chemist and I
can't do anything else.
No, especially young scientists.
Yes.
I think we often get scared, and maybe we even think that having a broad range of interest is not good.
Yes.
Because a successful scientist needs to be focused.
Exactly.
I agree, focus is important, but maybe we need to redefine what we mean by that.
Right.
And as long as we can focus on our broad interests, I don't see why that's a harmful thing.
Well, the other is that there's a recognition of complexity, and that there's very interesting that this shift is going on, like as we sit here. It's really amazing.
So one long-term extension of the work that I do would be to access the ancient cells, or maybe ancient behaviors. So we may never know what existed in the past. So this is maybe a shortage also to the approach that I'm taking, that I'm inferring what may have existed. The truth is we will never know what happened. It happened once, as far as we know, and that's it. So it got erased. That's one challenge in dealing with the past anyway, right? So history is written once. It is rewritten over and over again.
Yeah.
The geological record is scattered.
Right.
And unlike genome, the information that is secured by geological forces are not really driven by natural selection.
Yeah.
So the rocks that we see aren't here because they got selected somehow.
Yeah.
Usually it is more random geological scale
Even though
events.
There's a connection with biology. I mean, that these two affected each other.
Yeah, exactly.
Everything, but it's they don't have genes.
Exactly. So we think that maybe by reconstructing ancient pathways, by reconstructing proteins, who will change the behavior of the whole cell once we integrate them inside the genome, may provide us some insight and maybe help us to fill the gap, or maybe even attempt to fill this gap that we currently have in understanding the past.
Right. Well, you are certainly at the right time, it seems to me. Now, with good funding, you can accomplish a lot. Let's hope for that.
I've been very lucky that I've been funded very well in NASA, particularly exobiology and evolutionary biology programs, and NASA Astrobiology Institute, and NASA postdoctoral program has been funding my work. And lately, John Templeton Foundation very generously is funding my current activities here at Harvard.
Well, congratulations.
Thank you.
It's nice to hear that somebody is well funded, because so many people are really struggling, at this time. But in any case, the implications of the research, I mean, they're really wide, because you can open up the past in a more reliable way, I guess, reconstruct. And then there's the astrobiological thing. Are there any other implications that we should know about?
Well, there's always the underlying force that drives my research, which is understanding evolution.
Yeah, right.
That has always been at the heart. Understanding the basic mechanisms that drive adaptation.
Yeah.
How can we access those?
Right.
Currently existing methodologies, I think, offer a very wide range of, different variety of accessing information about evolution. But I see that integrating ancestral components and combining these components with modern cells could actually maybe provide a new and a complementary approach.
Right.
To
the system might change according to its environment. And is that possible?
So that's why, I think setting up systems where, experimental evolution, where we can evolve these populations in replication.
Yeah.
Is a good way of testing these, as you were saying, the inconsistency.
Yeah.
Right. So, for example, researchers like Michael Desai here at Harvard, or lately Johan Poulsen at Harvard Medical School, are all interested in setting up systems that will allow us to replicate even more bacterial populations than we did before. I think quantitatively improving our ability to study different bacterial organisms at large will be one way of testing the inconsistency in adaptation.
Right. Before we leave that, because we're going to ask you about how you got into all this in just a second as well, but in terms of, you're doing this in a lab.
Yes.
You've often, there are sometimes jokes about E. coli in the lab is not the E. coli out there in the wild. Is there any risk when you're doing this in the lab that the replication and so on might differ significantly from in the wild?
That's a very good question, and I just listened to a very nice talk by Andrew Murray, an evolutionary biologist here at Harvard, that asked the question of how much the evolution experiments that we do in the lab can inform what happens in natural life. And the idea is that we really want to perhaps access to this little tiny bit of detail of what is going on in nature, right? So the short answer is we can try. And is it actually replicating what's happening in nature? The answer is probably not. But it is far from what we want to achieve.
Right.
We are not achieving, our primary goal is not to necessarily replicate what is going on in nature.
Right.
We want to understand predefined mechanisms. We want to understand how gene adapts, how a population evolves with the parameters that we set up.
Right.
So, but I think with the new developments, again, in more technological developments, would allow our efforts to be more maybe closer to nature, if that's the goal of the researcher. Right. The truth is, we know very little even about what's going on in nature.
Well, that and you just pointed out, it's, your emphasis is the mechanisms themselves and the physical mechanisms that do that. So that makes a lot of sense. Plus, you have to control.
And there are studies like researchers in. We are in Boston, so I keep giving Boston references. But Roy Kishoni at Harvard Medical School do show beautiful examples of understanding adaptation of bacteria that were not necessarily evolving in the lab.
Right, not lab bacteria.
Not lab bacteria, wild bacteria that actually led to a complication in several patients. So there are beautiful examples now that shows us that what we develop through studying laboratory systems can definitely be applied to understanding the natural systems, because
Right.
we do develop models, we develop
Right,
different
right.
algorithms, we develop different systems,
Right.
and we understand fundamental mechanisms. There's definitely crosstalk between these two.
Yes, and evidently this modeling, the quantitative stuff is a very important part of it
Exactly.
too, right?
Yes.
That you are very careful with developing those
Yeah.
things, which gives you a huge insight
Exactly.
as well.
Yeah.
And I imagine that was probably not possible before you had computers
Yes.
to do this sort of thing. So the technology today allows.
Exactly, and my background is in chemistry, so I stumbled in this field because I fell in love with evolution.
Yeah.
But here, one of the reasons that why I am here is also that I can collaborate with researchers that develop these methods. So I'm
Right.
really looking forward to that aspect.
I bet. And now I would like to, before we close off here, I would like to find out how you got into this field. Give us a sense of your background, would you, and how you got into this very exciting area.
Well, my background, my Ph.D. was in chemistry, biomolecular chemistry. And when I was a graduate student, I been receiving a lot of books that were designed to explain that evolution is a lie, that evolution is not happening. And so I developed an interest in evolution through those books. I wanted to understand what can be so dangerous about an idea that created this kind of backlash. And I joined a team of outreach volunteers whose goal was to translate evolution documents in different languages. In my case, I'm from Turkey, so we translated these documents into Turkish. So through that, we translated the website of University of California, Berkeley. They have this beautiful understandingevolution.org website. We translated this whole website into Turkish and recorded this website as an offline form into CDs and distributed it throughout the nation. Through that, I learned about evolution, really, by translating. I learned about evolution, and I learned about experimental evolution. And that was it.
And that was it. You were sold.
I was
just
sold.
like
And
that.
then I started seeing more connections. I actually found more meaning in what I do as a chemist. I was studying proteins in yeast, but there was a big gap, really. I really think this is a big problem in some schools, that the graduate school programs do not incorporate evolution in fields like chemistry, biochemistry.
Not in biochemistry.
I mean, there is, of course, evolution, but it's from really the lens of maybe chemical evolution, really Darwinian selection, but not a fundamental evolution understanding was provided.
Very interesting.
And so not a understanding of populations, how populations evolve. And for a while, I was embarrassed about this lack of knowledge that I have, but then I realized...
You caught up, didn't you? That's for sure.
So wait a minute, maybe there's a bigger problem here. Yeah, and so I got I was really hooked.
Yeah.
The fact that we can evolve bacteria in the lab
Right.
and study the adaptation happening right in front of our eyes was mind-blowing to me. It's still
what
today.
an exciting thing, yes.
And I, that time, also was familiar with ancestral sequence reconstruction. Really exciting studies work. This was 2010. Really exciting stuff was coming out back then. And I joined a lab that primarily does ancestral sequence reconstruction. And I applied these ancient sequences into evolving systems. And so that's how it happened. So it's really from it.
Right.
Evolution is not true to,
Isn't that amazing? But it's a very encouraging story for other people, for young people coming up as well, from all over the world now. And you're familiar with this problem in the United States.
Very ironic.
So you think, yes, isn't it? And so that there is hope in that case, that people will do this, but you just went speeding ahead with all of this. Before we close off, also, I would like to ask you about, what is it?
Elsie?
Yes, thank you, right. Tell us about that, please, because that is really forward-looking.
Exactly. So apart from my research in the lab, I do outreach activities SaganNet is one of the example, and I also do activities that brings scientists together, that aims to bring scientists together. One of the, I would say, really game-changing happening in the field now is coming from Japan. Tokyo Institute.
Isn't that something?
It is wonderful. Tokyo Institute of Technology is has invested this. A lot of effort has been invested towards forming a center named Earth Life Science Institute, as we call ELSI. And this center has been endorsed by Japanese government. And really, like only, I think, 3 years ago, they came together and said, you know, we will solve the problem of origins of life. We want
Just
to
like
understand
that,
how
right.
life started. So from there, they brought together physicists and astrophysicists and chemists and biologists like myself, inviting us to Japan to give lectures, to give talks, just to sit in a room of people who are coming from different disciplines, as we were talking, who are interested in the similar questions, just discussions and really trying to form a framework, a team of people who will really go towards this question without the restriction of really worrying about grants and finding resources. So they did a tremendous job, I would say. It's been 3 years now, maybe 4, very young center. And they already have tens and tens of postdocs that are coming from different countries, a lot of different countries from Europe, from the United States. And they're all interested in understanding origins of life. And I'm one of their ambassadors, I would say, in the Earth Life Science Institute's Origins Network. So we are calling it as EON, ELSI Origins Network. The idea is to really create bridges between different fields, different scientists. So my job is to keep an eye on the ongoing research and find exciting areas and talk to young scientists and develop bridges between different fields towards origins.
That's great. I'm glad the Japanese are doing this. Somebody needs to be doing it, don't they? Because you need all of these different skills and people who aren't afraid to kind of get into things, but also it must be very stimulating to talk to people across disciplines.
It is very encouraging. It comes to me. I grew up with the books of Carl Sagan, and I am an astrobiologist because of him. Yes. Particularly the Contact book and later the movie really changed everything for me. And when I'm there, I always think of it as like, am I in a book of Carl Sagan now? It feels like a little bit out of this world, because just the nature of questions that we are asking, it is interesting. It feels out of this world, because if you think about it, it is actually out of I mean, it's actually inside of this world. How this world happened to be, and you know how life here started. So it's very encouraging also for a young scientist to see that. You know,
I think so, because it does. One of the downsides of being a young scientist is sometimes there are all those older scientists, those senior ones, and you feel like you can't breathe.
So there's hope.
Yes, good. Pass that
There's
along.
Hope, guys.
Because it seems like such an exciting time to be doing this. And I understand we have to close off here, but you do this outreach with the SaganNet. Also, I will put this up on the video so that people can go and check that out. I think we have it on your event page as well. But there may be some people out there that like to talk to you a little bit before we leave. And I thank you so much for joining us tonight.
Thanks for having me.
Very great fun to talk to you, and the best of luck.
Thanks. I will need it.
Okay. Good night.
Thank you.